Hello, ladies and gentlemen. I hope that you are all doing well. I hope that you are doing okay with genetics. Based on the work that I've seen, you guys are doing a really good job with handling this information. I'm so incredibly bummed that we don't get to go over this information in person, but this week we have a lot of really cool lessons kind of put together. We're going to talk about pedigrees and karyotypes today, and then tomorrow we're going to talk about genetic mutations and genetic disorders. All this week, you guys will be working on a genetics project. And I want to use that word project very loosely. This project is going to be a very small research-based project on a genetic disorder of your choosing. So before you dive right into that project, please make sure that you take a minute to definitely go through this lesson and the next lesson. That way you have a good background for the information that you're going to be looking up. So we're talking about pedigrees and karyotypes today. Let's get started. So before we go into really what pedigrees and karyotypes are, we need to review some vocab that we've seen before and some that we haven't. One of the words that we've definitely seen before are chromosomes. Chromosomes, thread-like structures that are made of DNA that contain genes, right? They're those X-looking chromosomes or those X-looking structures that we've been talking about since the beginning of this unit. Autosomes, we were introduced to last week in our X-linked trait lesson. Autosomes are any chromosome that is not a sex chromosome. So they're just our normal chromosomes that hold genes. There's not anything super special about them. Our sex-linked traits are traits that are controlled by genes located on our sex chromosomes. So X or Y chromosomes can hold certain traits that are specific to them only. And we kind of talked about that last week in the Khan Academy lesson. Karyotypes are a display of the chromosome pairs of a cell arranged by size and shape. So a karyotype just shows us what chromosomes look like in an organism, and pedigree charts can be used to track genetic information over several generations. So it's kind of like a family tree of how genes have been passed down, and we can predict how they will be based on our ancestors. So today we're talking about both karyotypes and pedigree charts, but this basic vocab is what we need to know moving forward, so that way karyotypes and pedigree charts make sense. So let's first focus on karyotypes, and I'm really excited about these. They're really, really cool to look at. They're very interesting, and hopefully you will find them as cool as I do. So a karyotype is the number and visual appearance of chromosomes in an individual organism. Now, it's basically just all of the chromosomes and what they look like. A lot of the karyotypes that we will be looking at are actually karyograms, so pictures of an organism's karyotype. Depending on the resource that you look at when you do research, the word karyotype and karyogram can be used interchangeably. So we're essentially talking about the same thing here. A karyotype looks like the picture that I have on the right. At the top right, we have your standard karyotype. You will mostly see black and white pictures of karyotypes or of an organism's genes or chromosomes. But every once in a while, we'll get a karyotype that looks like the bottom right, which is in color. Now, scientists and geneticists will assign different alleles, different colors, but that's just why it's in color, is because we have taken a picture of it and added color to it. It's not like we're glowing because we have radioactivity inside of us. That's not the way that it works. So a karyotype in the top right is what we're mostly going to see. Now we can learn a lot about an individual organism based off of their karyotype. We know that this karyotype in the top right is human because it shows us 22 autosomes and 2 sex chromosomes. Something important to note about autosomes. Autosomes come in pairs. So when we're talking about our 22 autosomes, we're talking about the 22 pairs of regular chromosomes. Chromosomes that hold genes but don't determine anything major like gender, right? Our two sex chromosomes determine what if we are male or female. Our autosomes just carry our DNA. Now, these 22 autosomes make up our 44 genes or alleles, and then those two sex chromosomes give us our 46 total genes. If we go back to meiosis and mitosis, every cell has to have 46 chromosomes. And so these chromosomes are what we see and what we're looking at in a karyotype. So when looking at a karyotype, numbers 1 through 22 are my normal autosomes. However, the last two, so 23 and 24, pair 23 and 24, are going to be those sex chromosomes. If we go back to the sex-linked traits Khan Academy lesson that we did last week, we learned that the sex chromosomes for a female are XX, and the sex chromosomes for a male are XY. Now, based on this karyotype, I will look at my X and Y chromosomes that I have here, and I notice that I have two X chromosomes. Based on that information, I know that I am looking at a karyotype of a female human. That human part is really, really important because it identifies what organism we're looking at. So, in addition to looking at this karyotype and determining the organism's sex, we can also look at a karyotype and determine different interesting facts about that organism. Now, if you look at all of the chromosomes, each chromosome looks like it's striped. And those different stripes that you are seeing are the alleles that we've been talking about the last couple of weeks. Now, every allele has a locus, and that locus is just a location of where that allele lives. And the cool thing about humans is that we know a lot about where the different alleles live. So for example, if I wanted to look at a karyotype and determine an individual's eye color, what I have to do first is I look at chromosome 15. Looking at chromosome 15 will tell me if that individual has a brown or blue gene for eye color. Once I look at chromosome 15, I then look at chromosome 19, and it tells me if that individual has a gene for blue or green eye color. And if they have a chromosome on 19, odds are, or excuse me, if they have an allele on chromosome 19, odds are they are a blue-eyed or green-eyed individual. People with brown eyes do not have alleles on chromosome 19 for eye color. They only have chromosomes on 15 because, again, brown eyes are a dominant trait. So these karyotypes can tell us a lot about the individual. However, karyotypes are most important for identifying genetic disorders. We are able to look at karyotypes and figure out and predict the likelihood of an organism passing down any genetic abnormalities, mutations, or disorders to the next generation. So for example, we're going to look at a couple of different karyotypes, and I'm going to kind of play this little game with you of spot the difference. But before we kind of get into that, I want to give you an example of what we're talking about here. So when I look at a karyotype of an individual with Down syndrome, I am looking at an individual who has an extra copy of chromosome 21. So remember back to the previous slide, we had all of our different chromosomes with a number associated to them, and chromosome 21 normally looks like this. However, in individuals who have Down syndrome, they have an extra copy of chromosome 21. That extra chromosome is what leads to different physical, mental disabilities for that individual. Now, the severity of those disabilities is dependent on that individual, but if we see 3 chromosomes at number 21, we call this trisomy 21, and that is the scientific name for Down syndrome. So these karyotypes can tell us a lot about one specific individual, but it can also tell us a ton, and we're able to actually look at different karyotypes and determine the different genetic disorders that that organism has. So let's practice this a little bit. So to show you what we were talking about when it comes to Down syndrome, this is what we are looking at here. In front of you, you see two different karyotypes. The one on the left is a karyotype of an individual who has Down syndrome, and the karyotype on the right is an individual who does not have Down syndrome. We know this, that the individual on the left has Down syndrome because of the extra copy of that chromosome at location 21. An individual who does not have Down syndrome does not have that. They have the normal 2 chromosomes at that location. And so when we look at different karyotypes, we can spot different abnormalities within the chromosomes, not just how the chromosomes look as far as bands go, but how many we have at each. Now, the next 2 slides are going to be karyotypes of individuals that have unique genetic disabilities, and I want to see if you can spot the difference between the two. So here are 2 karyotypes. The first thing that you should do anytime that you look at a karyotype is you should count the number of autosomes or normal chromosomes, nothing special fancy chromosomes, and see if there's any differences in them. So I have 1 through 22 autosomes. I see that each pair of autosomes only has 2 chromosomes, so that rules out Down syndrome automatically. I looked at the picture at the right, and I see that chromosomes 1 through 22 are the same. So between left and right, those 2 chromosomes in the autosome chromosomes are the same. So there's nothing different there. The next thing that I should do when looking at a karyotype is looking at the sex chromosomes. So I can look at pairs 23 and 24, and automatically on the left side, I notice something interesting. So before we get there, the one on the right, we have 1 chromosome that is X, 1 chromosome that is Y. Therefore, we know that we are looking at a male. But the karyotype on the left has something interesting. We have 2 X chromosomes and a Y chromosome. This is what we call Klinefelter syndrome. Klinefelter syndrome is when a male individual is born with 2 X chromosomes in their sex chromosomes. And this causes a lot of physical abnormalities, a lot of developmental or puberty problems for that male that they will experience for the rest of their life. So it is very severe in what it does, in that they don't develop the same way that a normal male would. So now that you kind of know how to approach a karyotype, I want to give you some time to kind of look through these 2 karyotypes that you have in front of you, the one on the left and the one on the right. Go through the autosomes, go through those sex chromosomes, and see what difference you can find. So now that you've gone through the 2 of them, let me show you what I see. After looking through my 22 pairs of autosomes, I don't see any difference between the 2. I know that I am looking at a female on the right side. So this has to be my normal female because she has 2 X chromosomes. However, the one on the left is where this abnormality is different. On the left side, we have the karyotype of someone that has Turner syndrome. Turner syndrome individuals are females that are missing that second X chromosome. So it doesn't exist. Much like males that have Klinefelter syndrome, females with Turner syndrome experience severe differences in their development as females. There's a lot of physical abnormalities, there's a lot of stunted growth because of this chromosomal abnormality. So these karyotypes can tell us a lot about individuals and what they, not only the genes that they have, but what they are as a whole. And we're able to see those genetic disorders within people who have genetic disorders. So the next thing that we're going to talk about are pedigrees. And pedigrees are very similar to karyotypes in that it tells us a lot about a person's genetics, but pedigrees are at a bigger scale. So pedigree charts are what you see on the left. Those shapes, those lines, all of those things mean something. That chart is showing us a family across multiple generations, and based on whether or not that shape is filled in or not, we can tell what individual has what trait. So think of a pedigree chart kind of like a family tree of inheritance patterns. Based on what an individual has in generation 1, they will pass that down to generation 2, and they will then pass that down to generation 3. Every pedigree chart is going to be read the same. So a circle represents a female, a square represents a male. Any circle or square that is shaded in means that individual is affected with whatever trait we are looking at. If that circle or square has a line going through it, that means that individual has passed away. If the circle, square, whatever it is, descend from the same line, they are twins. If they are in parentheses, they have no genetic similarities due to being adopted. And if it is a triangle, that means that individual had a miscarriage. Now, the next thing that we do when we go to read a pedigree is we determine if the trait is dominant or recessive based on the inheritance pattern that we see in the previous generations. We then determine if that chart shows an autosomal trait, meaning a trait carried by one of those 22 pairs of chromosomes, or if it is sex-linked, meaning that trait is carried on the X or the Y. Usually that information is given to you just in background information, but every once in a while you can come across different questions that ask you to conclude whether or not that trait is dominant, recessive, X-linked, or autosomal. So the whole reason that we use pedigrees in the first place is because we can predict the chances of certain genes being passed down to the next generation. And this is most helpful when it comes to our sex-linked or X-linked traits like color blindness. Now, based on a family's history, we can conclude whether or not the upcoming generation will be or have that gene. So for example, color blindness is a trait that is X-linked. It is passed down on that X chromosome. Now, it is more common in men than it is in women solely because men only have one of those X chromosomes. It's a recessive trait. Therefore, you need to have at least two copies of that trait. So if a female were to be X chromosome for colorblind with another X, she is a carrier, but she herself is not colorblind. A female can only be colorblind if she has both X chromosomes affected by color blindness. However, let's go back to this male. Now, our male is colorblind, and based on the woman that the women that he has in his family and the woman that he ends up being with to produce offspring, we can predict his chances of having children who are also colorblind. So let's take a look at kind of what we're talking about here. So this key here, this visual, is kind of a layout and some basic reasoning and some basic logic behind how pedigrees work and the information that a pedigree can tell us. We're going to walk through two examples, but I wanted to mention that there are four different ways that a pedigree can work. They can either show us autosomal dominant traits, autosomal recessive traits, X-linked traits, X-linked dominant traits or X-linked recessive traits. So as you work through the next two slides that we're going to walk through, I want you to kind of think about the information that you have in front of you, and you can refer back to this slide in our video as we move forward. So this first example that we're going to walk through is of an autosomal dominant trait of just having freckles. So it's literally whether or not you have freckles as an individual. It has nothing to do with sex chromosomes. It's literally just what do you look like? So we know that the allele for freckles is a dominant trait. We know this because it's given to us in our background information here. Therefore, the individuals who have freckles have that phenotype because it is a dominant trait. Again, the law of dominance. If you have at least one allele in your genotype that is dominant, you will have freckles. Affected individuals will be filled in like so here. Any individual that has freckles is going to be filled in with black. Now, this shows us 3 generations, and we know this because we literally have 1, 2, and 3 put right to the left of this picture. So your next question has to be, how do we know that this is dominant? Well, we know that individuals who have it are going to show it. So we have a person in generation 2 that has freckles here. Of their offspring, which stems from this line here, 2 of the 3 offspring have freckles. However, one of them does not because one of the parents does not. We can assume that parent 1 is recessive, so lowercase d, lowercase d. And we can assume that parent 2 is going to be heterozygous. Now, why can we assume this? Well, we have individuals who don't have that trait. In order for us to have a recessive trait show up, we need to have 2 recessive alleles in their genotype. However, it is more frequently occurring that freckles show up. So we know that this trait is autosomal dominant based on the inheritance pattern that this family displays. Now, this next example is of a sex-linked recessive trait for being colorblind. Based on the background information, we know that being colorblind is a recessive trait. Therefore, it isn't always guaranteed to show up. What we notice is that in generation 1, neither parent has that trait. However, in generation 2, when these 2 individuals mate and produce offspring, they have one individual who is affected by colorblindness. This means that one of the 2 parents is a carrier. And even further so, we know for 100% fact that carrier is mom. Think about it. If mom was a carrier, she has one X chromosome that is affected. If dad was colorblind, he would have his shape filled in here, but he doesn't. He does not have colorblindness, but mom does. So he or they together are likely to pass down colorblindness to their offspring. Now, again, it's recessive, which means it's not likely to show up unless this individual, who again, note, squares are male, this individual only has one X chromosome, meaning that because his mom was a carrier, otherwise known as XCX, he inherited the genotype XCY. And this is the only genotype. And if you notice throughout here, we have all male individuals with colorblindness. It is because it is more likely for males to be colorblind than women. In order for women to be colorblind, they need to have XC. And it's just not very likely that this is ever going to happen. So the good news about karyotypes and pedigrees with the information that we just kind of walked through is that you need to be familiar with this stuff. By no means do you need to be an expert in this area. So I've put together this slide of what you need to know. With karyotypes, you need to know what a karyotype is. A karyotype is just a layout of all of the chromosomes and what they look like. It's an actual picture of what they look like. Based on that picture, we can determine different chromosomal abnormalities that can show up. We can see if a person has 21 chromosomes, or excuse me, if a person has 3 chromosomes at chromosome 21. And if they do, we know that they have Down syndrome. We also need to know how to read it. We need to know that there are 22 autosomes that show up and 2 sex chromosomes that show up. If you have 2 chromosomes at X, you have a female. If you have 1 chromosome at X and 1 chromosome at Y, you know that you have a male. With pedigrees, the only thing that you really need to know is that a pedigree chart lays out the genetic history of a family. And based off of the genetic inheritance of that family, we can predict the chances of offspring developing whatever that gene is. Based on a pedigree, we know whether or not an offspring will have color blindness. So ladies and gentlemen, if at any point you are struggling with any of this information or you're just interested in learning some more, I've linked a couple of websites and resources for you in the description of this assignment. I highly encourage you to go check it out, especially if there's a little bit of confusion with pedigrees or karyotypes. As always, you are always welcome to reach out to me with any questions, comments, or concerns that you have. Please let me know how I can help you. Please let me know how I can support you. You have a big project coming up this week and due at the end of next week. So reach out with questions for that. Make sure that you listen to tomorrow's lesson. And ladies and gentlemen, as always, have a great day, be safe, and make good choices.